Hello, welcome. This is a video on, well, just sort of an overview of what you're going to be doing for lab experiment number 3. It does cover mitosis and meiosis. So I thought I would go through and show you, just give you an overview of what you're doing. I think the instructions are pretty clear, but I did want to show you a couple of things. So here is our class. I'm going to go down to week 3. There we are. Here is the lab. And here we go. We got 2 experiments that we're doing. Now remember, always check the due date, the My Grades tab for due dates. The due dates for these experiments are different than other assignments. You have a little bit more time. All right, lab number 3. Now here is the answer sheet. I'll get to that in a second. So what you're going to do, click on this. I gave you a chart here. It's pretty easy to follow. It's over meiosis. We don't cover that a lot in anatomy and physiology, but you guys are going to be doing a little demonstration with it. Meiosis refers to division of sex cells, not somatic cells. So I'm going to click on the link here for the eScience. Now, I've said this before. You can go over here and click on New Window, make this big. Now, I really like these little modules they have. I think they're easy to read. You're going to go through, obviously, and read through. There's actually a check here. So what was it? Knowledge check. Which phase of the cell cycle does the cell spend the majority of its time in? And that would be interphase. But anyway, here's a nice little breakdown of mitosis. Read through that. I think it's good. This is called active learning, and it's pretty straightforward. There are only 4 stages of mitosis. Mitosis is division of non-sex cells, or what are called somatic cells. Every cell in your body except for sex cells. So if you're a guy, sperm cells divide by meiosis. If you're a female, egg cells divide by meiosis also. So mitosis involves just what are called the somatic cells, the non-sex cells. Every cell in your body but sex cells. So you will read through. It gives you a nice little demonstration on animal cells here. I've already looked at these. I really like this. There was a way to make this full screen. Let me go home. Well, I'm sorry, there it is. Sure, why not? Anyway, here you go. So it gives you a nice little breakdown. See, it tells you what happens during each stage. So go through this module. When you get done, now I would do the little quiz here at the end. That's great. I like the way they do it. It's a good recall thing for you. But assignment and procedures. Now, here's the part. I changed this up just a little bit. So I'm going to go back out. I'm just going to go backwards. One more click. And here is the lab answer sheet. I took off some of the parts for this particular exercise. In any lab assignment, I only want you to do what is posted on the lab sheet. All right? That module may cover a few extra things I want you to do here, and this is one of these you're going to have to use pictures for. So go down here, some pre-lab questions. What are chromosomes made of? Long strands of chromatin wrapped around histones. You can research that yourself. Questions like this. In fact, I clicked on this. I need to give you a point value. But these pretty easy. Do a good job. Do a little bit of thorough research. Answer your questions. And you should get full credit. Go ahead and answer these 4. And some of these like this. Use the knowledge to invent a drug that prevent the growth of cancer cells. Look, you can't really get that wrong. You can't put, Fruit Loops there or anything. And if you don't do it, you'll get it wrong. But if you put forth an effort, hey, I think this is what would happen if I created a drug that would block either oncode genes or tumor suppressor genes or activate tumor suppressor genes, just have some fun with that. Now down here, for instance, like this, the predictions, the supporting evidence, really can't get that wrong. However, I'd like you to write some thoughts here. If you don't put anything, you'll get it wrong. Here are the stages of mitosis. You will go through, you'll see this in the assignment portion. Let me click on this. So let me go back. I thought I already had this open. There it is. Observations of a plant cell, and it even says here is the slide viewer. So you can go through and you will look through all of these onion tip slides. These are all in different stages of mitosis. Okay, you do this in lab. So there's a nice little telophase right there. There's a metaphase. Some of these just kind of scanning around. That looks like a prophase. Can't really get that wrong either. All right, and you're gonna do a little calculation here. It shows you in the module how to do it, and you'll just fill in your data here. Now, post-lab questions, look at this diagram, put what stages you think those are, do a good effort, you'll get full credit, okay? And then answer these questions. I need to give you a point value here. I don't know why I posted the wrong one. Now right here, you're going to post a picture. What you're gonna do is in your lab kit, you have some beads, you're gonna connect them together and follow the instructions. Now look on this document, when you do that, it really kind of spaces this out a little bit. But I've already gone through and done this experiment. I'm gonna pick the one for prophase, and obviously I'm gonna make it a little smaller. What you're supposed to do, and look, you can do these, you can do the little position thing. It's kind of iffy. Middle, anyway. Well, there it is. And you can, if you want to, you can make this go up a little bit more of it. So there it is. There's a nice little, look, look what I did. That to me is, chromosomes condense. They are chromatin fibers, and then they condense during prophase, the first stage of mitosis. You cannot see chromatin fibers in a nucleus. They're dissolved. They're really long. One of the reasons that chromosomes condense is they're too long as chromatin fibers. But when they condense, you can see them as dark stains in the nucleus. That's Greg's impression of dark stained chromosomes in the nucleus. And yes, they do double up like this. You have what are called sister chromatids. So you're going to make these. It shows you how. There's some instructions. You're going to align them for metaphase, which they line up in the center. You're going to do one for anaphase, one for telophase. And then one for cytokinesis when they're splitting apart. All right. You're then going to answer these questions. And then this is meiosis. And I did pick, which was prophase 1. Meiosis has 2 divisions to it. You end up with half the number of chromosomes and a little bit of genetic variability. There's actually 2 prophases, 2 metaphases. But it says, do a B diagram without crossing over. And, well, I could probably I want you to fill these in. I took a few pictures. Here is, like, for instance, that. Well, that's the wrong one. Meathead. Here we go. There we go. There's a nice B diagram that does not involve crossing over. Crossing over is where the chromosomes actually touch and they exchange genetic material. You can see these are the same, or what are called homologous. But you're going to arrange them into a meiosis type division and put your pictures here. And then do one cell cycle with crossing over. And that's where little pieces break off, and then you're going to answer these questions. That's the entire lab. To be honest, if you wanted to, you could sit down and do this easily in a couple hours. No problem. I need to add some point values to this for you. One real quick look at, if you want to know what that means by crossing over, that was my original one. Chromosomes touch during meiosis, and when they do, you end up with little pieces breaking off and they attach. That's where the genetic variability comes in. And if you're going to line them up like that, see, I need you to do something like this. And I did give you a diagram that's pretty easy to follow. You go back out to our, right here under our experiment. Yeah, there's one section where it's referring to this is not crossing over, this involves crossing over. So if you'll line them up sort of like this. Okay, and then down here you have what are called the actual daughter cells, the offspring of the cells. And you can see they are haploid, which means they only have 23. You'll get to that. But that's experiment 3. I hope this helps. It tells you pretty detailed, but I do want you just to answer the questions that I have posted on this answer sheet. And after this, you only have one more experiment as far as the first section of this class. And this is the majority of the experiments you're going to do. You're going to do something a little later. It might take you some time, but at the moment, there's only 5 of these. So you will have done almost the first 4 within the first part of this class. So this is number 3. Okay, well, have a good night. And like I said, you should be able to do this easily within a couple of hours.